Steam start. Good morning, students. Today I am going to explain you enzymes. So already in the previous class of this biochemistry, we have started with the enzymes, and you know that enzymes are proteins which have catalytic activity. Those we can call it as enzymes, and you know the role of the enzymes because each single biochemical reaction has been catalyzed by enzymes. Okay, see here. Enzymes are nothing but proteins, few proteins which has catalytic activity. So, what you disturbance? Yeah. See here, enzymes which are nothing but proteins which has like catalytic activity. So those proteins which has catalytic activity, we can call it as enzymes. And you know that biochemical reactions, in order to derive energy from the food has to undergo biochemical reactions. And a single reaction without takes place without the enzyme. So enzymes which catalyze the reaction, they act as a biocatalyst. They will speed up the reaction without undergoing any change during the biochemical reaction. And you know that many enzymes are there. All the enzymes have been classified into 6 classes that already we have discussed in the previous class. So the major 6 classes are like first one, you should follow the order. That is, the first one is oxidoreductase, transferases, then hydrolases, lyases, isomerases and ligases. So these individual enzyme action already with the examples we have finished in the last class. Now coming to the factors which will affect the rate of the enzyme action. Okay, so that means the rate of the reaction. What is the enzymatic reaction? Enzyme plus substrate, that will give rise to enzyme substrate complex. Okay, see here, enzyme that will bind with the substrate to form a complex, that is enzyme substrate complex. See here, this enzyme substrate complex gives rise to enzyme plus products. So that means enzyme is utilized in the reaction and at the end it is going to release. But meanwhile what will happen? Substrates are going to convert it to products. So enzymatic reaction is enzyme plus substrate gives rise to enzyme substrate complex, that is ES complex, which is further converted to enzyme plus products. So this rate of the enzyme activity depends upon the many factors. So the main factors which will affect the rate of the enzyme activity is that depends upon the concentration of the enzymes, concentration of the substrate, and the temperature of the surroundings, and even the pH of the medium, and effect of the product concentration, effect of the light and radiation. Okay, right here this rate of the enzyme action. Rate of the enzyme action means I have already mentioned enzyme plus substrate gives rise to enzyme substrate complex. Okay, always substrate will go and bind to the enzyme. Enzyme has a specific site on the structure for the binding of substrate. Enzyme has a specific site for the binding of the substrate. So substrate will go and bind to the enzyme to form enzyme substrate complex. So the binding of the substrate to the enzyme causes the catalysis that will convert the substrate into products. Again enzyme is going to release. So this rate of the enzyme activity, it mainly depends upon the concentration of the enzymes, concentration of the substrate, effect of the temperature, okay, and it depends upon the pH of the medium, and even the products also, they will have influence on the enzyme activity, and even exposure to the light and radiation also, they will affect the rate of the enzyme action. Okay, see here the contact between the, so for a biochemical reaction to takes place, what is the essential one? First, enzyme should contact with the substrate. That is the most essential prerequisite for enzyme activity. Okay, so enzyme to exhibit its function, First, enzyme has to bind with the substrate. The important factors that influences the velocity, that is the rate of the enzyme reaction, is discussed here under. So next, first let us see the effect of the temperature. So how the temperature will affect the rate of the enzyme action. So enzyme is the most active at the specific temperature, which is called optimum temperature. See here, the temperature. Now what we are discussing, how the factors, different factors will influence the rate of the enzyme action. Okay, so first we are discussing about the temperature. Each enzyme has got means most of the enzymes are specific at particular temperature. That temperature is known as optimum temperature. Okay, so that means at that temperature the enzyme exhibits its maximum action. Okay, and the maximum optimum temperature is our human body temperature, that is 37 degrees centigrade. 37 degrees centigrade is the optimum temperature for the activity of the enzymes. So temperature increases the total energy of the chemical system. So with the result of activation energy is increased. So the exact ratio by which the velocity changes is 10 degrees centigrade. Temperature rises to 10 degrees of the coefficient. Okay, see here what you have to understand is temperature has an influence and the rate of the enzyme action right here. And what is the action of the temperature on enzyme activity means, as the temperature increases, what will happen? There is a total energy of the chemical system is increased. So that increases the activation energy. So that's what, when you increase the temperature, slowly the rate of the reaction is increasing up to which degree centigrade? 37 degrees centigrade, because that is the optimum temperature. After that, further you increase the temperature, there is decrease in the rate of the reaction. Why? Because beyond that temperature proteins are denatured. Okay, proteins are denatured, they loses its like biochemical catalytic activity, and there is a decline in the rate of the reaction. Okay, see here, so that's what, when you draw a graph between, when you plot a graph between the temperature with the enzyme activity, a bell-shaped curve is obtained. So that means most of the enzymes of human system have an optimum temperature, mostly between 35 to 40 degrees centigrade. Above or below the optimum temperature, there is decrease in the enzymatic activity. Okay, look at this picture. This is the picture which is showing the temperature, effect of the temperature on enzymatic reaction. On x-axis we have taken the temperature, and y-axis the rate of the enzyme activity. Suppose first you are increasing the temperature up to 37 degrees centigrade. What will, so this is the peak point. What is this peak point? That is called the optimum temperature. Okay, this peak point is called the optimum temperature. So this is the maximum activity of the enzyme, right? So this is the rate of the enzyme activity we have recorded at different temperatures. Right, see here, slowly if you increase the temperature, the rate of the enzyme activity is increasing and it reaches the peak at certain temperature. After that there is a decline in the temperature. So this peak point, this is the maximum enzyme activity at which temperature? That is in between 37 degrees centigrade. That you can call it as optimum temperature, right? Optimum temperature means the temperature at which the enzyme exhibits its maximum activity. Below or above, okay, below the optimum temperature, right, and above the optimum temperature, if you increase or decrease, there is a decline in the rate of the reaction. So that's what, when you draw enzyme activity versus temperature, there is a bell-shaped curve is obtained. Okay, a bell-shaped curve is obtained if you draw a graph, if you plot a graph between the enzyme activity and increasing the temperature. Then coming to the next one is effect of the pH. The rate of the enzymatic reaction also depends upon the pH of the medium, right? The enzyme activity is maximum at a particular pH, which is called optimum pH. Okay, right, that first we have discussed about the temperature activity. So a bell-shaped curve is obtained because at certain temperature the enzyme activity is maximum. Below or above, due to the denaturation of the protein enzymes, there is decline in this one. And then coming to the next one is effect of the pH. pH also influences the rate of the reaction. So the enzyme activity is maximum at a particular pH, which is called optimum pH, right? The optimum pH of the most of the enzymes lies between, mostly it is our body temperature, that is 7.4. okay, the optimum pH for most of the enzyme is 7.4. any increase or decrease of the pH decreases the rate of the reaction. Okay, see here, and this is also the graph. See, look at this picture. This is the graph which is plotted between the rate of the enzyme activity on y-axis and increase of the pH on x-axis, right here. So this is the increase in the pH. So here at the neutral pH, almost 7.4, the enzyme activity is maximum. Okay, enzyme activity, the rate of the reaction is maximum at the pH 7.4, since that is our body pH. So that pH we can call it as optimum pH. What is the definition of optimum pH? The pH at which the rate of the reaction is maximum. Any increase or decrease of the pH above or below the optimum pH, there is slowly decrease in the enzymatic reaction. That's what, if you plot a graph between pH and enzyme activity, a bell-shaped curve is obtained. Then coming to the next one, effect of the enzyme concentration. Enzyme concentration means even the rate of the velocity of the reaction, it depends upon the enzyme concentration. Okay, in the beginning the velocity of the enzyme reaction, if you increase in the enzymatic reaction, what will happen? There is increase in the rate of the reaction. Okay, if sufficient substrates are there in the medium, if we increasing the enzyme concentration, the rate of the reaction is also increasing. But when the substrate concentration is large, exceeding that of the Vmax, because enzyme is the limiting factor in the enzyme substrate reaction, and providing more enzyme molecules enables the conversion of progress large molecules of the substrate molecules, right here. And if you plot a graph, okay, this is the graph between this is the enzyme concentration, this is the rate of the reaction. Okay, if sufficient substrates are there, okay, if sufficient substrates are there, then the increasing in the rate of the enzyme, okay, increasing the concentration of the enzyme, increasing the rate of the reaction. That's what a straight line, linear line, okay, straight line is obtained if you plot a graph between the velocity of the reaction versus enzyme concentration. Okay, so increasing in the enzyme velocity of the reaction is directly proportional to enzyme concentration if sufficient substrates are present. Okay, that you have to mention. If sufficient substrates are present, then the rate of the reaction, the velocity of the reaction is directly proportional to enzyme concentration. Means increasing in the enzyme concentration, simultaneously increasing the rate of the enzyme action. Then coming to the effect of the product concentration. If excess products are there, what will happen? The products itself go and inhibits the enzyme. So products which are formed as a result of that enzymatic reaction, if they accumulate and excess is there, that will decreases the rate of the enzyme action. Okay, and it is also possible that under certain conditions of high concentration of the product, a reverse reaction may be forming the back of the substrate. Sometimes products, usually if sufficient products are there in the medium, the product itself decreases the velocity of the reaction. Sometimes, rarely, products may converted back to substrates. So that's what, like that, the velocity of the reaction, that is the rate of the reaction, depends upon the temperature. If you graph, what is the shape of the temperature? Bell-shaped curve, and it depends upon the pH. Optimum pH for the action of the enzyme is 7.4, and the graph, the shape of the graph is bell-shaped curve. And the next one, it also depends upon the enzyme concentration. What is the shape of the enzyme concentration? If you plot a graph between the this substrate concentration versus velocity of the reaction, there is a linear line, straight line is obtained. And even the products which are formed in that reaction also influences the velocity of the reaction. If more products are there, that decreases the rate of the reaction, and moreover sometimes products may be converted back to substrates. Then coming to the effect of the substrate concentration, even the availability of the substrate also influences the enzyme activity. Okay, so first you take a test tube, series of test tubes, equal volume of the enzymes has been added. Okay, take 5 test tubes, in each then add 1 ml of the enzymes. Then what you have to do is increase the quantity of the substrate. Add 1 ml of the substrate to the first test tube, 2 ml of the substrate to all the 5 test tubes. You have added equal concentration of the enzymes. But later the substrates you are increasing. In the first test tube you have added 1 ml of the substrate. In the second test tube you have added 2 ml of the substrate. Like that you are serially increasing the concentration of the substrate, and then find out the rate of the reaction. Okay, if the velocity of the reaction is plotted against the substrate concentration, a typical different curve is obtained, that is a rectangular hyperbola. Okay, this is the shape of the curve. So this is an essay question for you. Okay, classification of the enzyme is an essay question for 10 marks, and even the what we are discussing now, factors affecting enzyme action, also important essay question for you. If the question is asked for an essay, you have to write about all the factors and you should draw the graphs also. So this is the graph which is drawn, which is plotted between the velocity of the reaction on y-axis versus like increasing the substrate concentration on x-axis. So what is the shape of this graph? This is rectangular hyperbola. So that means why it is rectangular hyperbola means initially what happened, sufficient enzyme is there, so for the substrate to react. So that's what, if we increasing the substrate concentration, as long as the enzymes are available, the rate of the reaction is directly proportional. Right here already enzyme is there, nicely you are adding the substrate, so enzyme and substrate will react to form products. So initially the rate of the reaction increasing in the substrate concentration, increasing the rate of the reaction. Okay, the velocity of the reaction is directly proportional to increasing in the substrate concentration initially. After that what will happen, all the enzymes enzymes are limited only, but still you are adding more substrate. So after that still you are increasing the substrate concentration, there is no more enzymes to act. Okay, you are increasing the substrate concentration, right, but there is no more enzymes to act because already all the enzymes have been used. So that's what, if further increasing in the substrate concentration, there is no increasing in the velocity of the reaction. Reaction will not occur. Why the reaction will not occur? Only enzymes, all the enzymes which are you have added already finished with the substrate, but still you are increasing in the substrate concentration, but simultaneously you are not increasing the enzyme concentration. So that's what, after some time what will happen, the velocity of the reaction is not depend on the substrate concentration. Okay, that's what, if you plot a graph between the velocity of the reaction versus the substrate concentration, a rectangular hyperbola is obtained. Why rectangular hyperbola means initially the rate of the reaction is directly proportional to the substrate concentration, but later on the rate of the reaction is independent on the substrate concentration, because why, you are increasing the substrate concentration but enzymes are not there. Okay, so this is the one, and the next one is here order of this. Okay, finally this is the reaction. Okay, enzymatic reaction is enzyme plus substrate to form enzyme substrate complex, which is further catalyzed to product plus enzyme. So what is the role of an enzyme in a biochemical reaction? It just acts as a catalyst. Catalyst role means it increases this, it speed up the rate of the reaction, but it will not involve in the chemical reaction. Okay, as long as the we are discussing about the enzymes, you should remember this equation. Enzyme plus substrate gives rise to enzyme substrate complex. Enzyme substrate complex further gives rise to product plus enzymes. Now here, so these are the main factors which will affect the rate of the enzyme action. Okay, till now what we have discussed: effect of the temperature, bell-shaped curve; effect of the pH, bell-shaped curve; effect of the enzyme concentration, a straight line; and the effect of the product concentration, which will decreases the rate of the reaction. And effect of the substrate concentration, the shape of the graph is rectangular hyperbola is obtained. And one more factor which also influences the rate of the reaction is effect of activators and coenzymes. Okay, the activity of the certain enzymes is mainly depends upon the, because already you know that enzymes for their fully functional they should have the along with the protein part they should also need the non-protein part. In the previous class we have discussed holoenzyme is equal to enzyme plus apoenzyme. Okay, that means in the full activity of the enzymes, two should be there, that is protein part should be there and non-protein part should be there, then only enzyme is completely active. The non-protein part may be a coenzyme, okay, or it may be a metals, or maybe a biochemical, means biochemical activators of the B complex vitamins. Okay, suppose if those activators, if there is a B complex deficiency, what will happen? That will inactivate the enzyme, because that B complex active form is needed for the particular enzymes to act. If B complex deficiency is there, that will inactivate the enzyme. So that means the rate of the reaction, it also depends upon the enzyme activity. Enzyme activity in turn depends upon the availability of the activators and the coenzymes. Okay, that's it about this one. For example, you take the enzyme that is pyruvate oxidase. This is the one of the enzyme. Okay, pyruvate oxidase. This is the one of the enzyme. For its complete activity, it requires the metal that is manganese. For example, you take phenol oxidase. It is completely active when copper is present. Suppose if copper deficiency is there, that inactivates enzyme phenol oxidase. Okay, like that. And lastly, even there is effect of the time also. Time also influences the rate of the reaction. So the time required for completion of an enzyme reaction that increases with the decrease in the temperature from its optimum. Okay, suppose if the temperature is low in that medium, obviously the time taking for the enzymatic reaction to take place is increasing. Okay, however, so under the optimum conditions of the pH and temperature, time is required for enzymatic reactions less. Okay, suppose if you provide the optimum pH and optimum temperature, the time required for the chemical reaction is decreasing. Okay, suppose if optimum temperature is not maintained, then obviously there is increasing in the time for the reaction to takes place. Okay, so like that, these are all the main factors which will affect the rate of the enzyme action. So that is effect of the temperature, pH, and effect of the time, effect of the metals, metallofactors and coenzymes, and substrate concentration, and the enzyme concentration and product concentration. 7 you have to mention. For the main 4 you have to draw the plots. For the temperature, for the pH you have to draw the plot, bell shaped curve. For the substrate concentration, rectangular hyperbola, and for the enzyme concentration, linear. Okay, that's it about the rate of the enzyme action. Then we'll move on to the next topic of the enzymes. Okay, give me 1 minute time, I'll change the slides. Okay, next we will move on to the next topic of the enzyme, that is enzyme inhibition. Because if the enzymes are always active, they should be active whenever its need is there. Other times they should be shut down. So that means enzyme inhibition is nothing but regulation of the enzyme activity. Okay, see, enzyme inhibition means it's a process of decreasing, okay, interference with the catalytic activity of the enzymes by the use of inhibitor molecules. Okay, inhibition means the enzyme inhibition is defined as the process of decreasing the enzyme activity. Okay, the process of decreasing the enzyme activity is known as enzyme inhibition. Then enzyme reaction is slowed down. Okay, in the presence of the substance which causes the enzyme inhibition is called inhibitor. So the enzyme reaction is slowed, or in the presence of the inhibitor the enzyme reaction is slowed or halted. So what is an inhibitor? Inhibitor means enzyme inhibitors are the substances which bind with the enzymes and bring about a decrease in the catalytic activity of the enzyme. Okay, first you should know what is enzyme inhibition. It's a process of interference, that means decreasing the catalytic activity of the enzyme by the use of inhibitor. Okay, first you should know the definition of the enzyme inhibition properly. That is, it's a process of decreasing the enzyme activity by using the inhibitors. The enzymatic reaction in the presence of inhibitor is obviously it will stop or slow down. Then what is an inhibitor? Inhibitor means these are the substances which bind with the enzyme and bring about a decrease in the catalytic activity of the enzymes. Right here, inhibitor is responsible for the enzyme inhibition. Then what is an inhibitor? It's a substance. It binds with the enzymes and decreases the catalytic activity of the enzymes. Okay, enzyme inhibitors are mainly used to gain the information about the shape of the active site and the amino acid residues at the active site. Okay, what is an active site? The substrate will go and bind to specific site on an enzyme. That site you can call it as active site. Okay, so here I told the equation, no, enzyme plus substrate gives rise to enzyme substrate complex, that in turn gives rise to enzyme plus product. Right here, so the substrate which binds to the specific site on the enzyme, that site you can call it as active site. Okay, so enzyme inhibitors are mainly used to know the shape of the active site and to know which amino acids are there in the active site. And it also gives the information about the mechanism of enzyme catalysis, how the reaction takes place. And it also gives the information about the metabolic pathway, and it can be very important in drug designing. Okay, so now please note down why the enzyme inhibitors are used. Okay, and enzyme inhibitor means these are the inhibitors, these are the substances bind to the enzyme and slow the rate of the reaction. So what is the use of this enzyme inhibitor means to understand the metabolic pathway, to understand the drug designing, and to know the mechanism of enzyme catalysis, and also to know the shape of the active site and amino acid residues. Okay, and most important, pharmaceutical drugs. Okay, mainly the drugs what we are taking to control the various ailments, diseases, are mainly the enzyme inhibitors. Okay, most of the drugs, their mechanism of action is they may mainly inhibit the enzyme activity, thereby controlling the diseases. Okay, please note down, most important pharmaceutical drugs are enzyme inhibitors. Right, now coming to the enzyme inhibition, it is of two types, reversible and irreversible. Reversible means, so first what is the reaction? Enzyme plus substrate gives rise to enzyme substrate complex. Enzyme substrate complex gives rise to enzyme plus products. That reaction I think you know. Okay, I repeat, what is the chemical reaction? Enzyme plus substrate gives enzyme ES complex, that is enzyme substrate complex, which gives rise to enzyme plus inhibitor, enzyme plus product. So inhibition, now this is the rate of the reaction. Now we are discussing about the inhibitors. What are the inhibitors? These are the substances which will bind with the enzyme, rather than actually who has to go and bind to the enzyme? Substrate. But here what happened? In the place of substrate, okay, rather than substrate, the inhibitor will go and bind to the enzyme, thereby decreasing the formation of the products. Okay, so that process is known as enzyme inhibition. This enzyme inhibition is of two types, reversible and irreversible inhibition. Okay, so the inhibition can be classified. Inhibition is nothing but, enzyme inhibition is nothing but the process in which the enzyme activity is decreased. So that process can be of two types, reversible, irreversible. Reversible means here this type of, by removing the inhibitor, this type of inhibition is reversed, enzyme coming to the normal state. But irreversible means here you can't remove the inhibitor from the enzyme. So once the inhibitor binds to the enzyme, there is no more enzyme coming back. Okay. So enzyme inhibition is of two types: reversible, irreversible. Under reversible also again three types are there: competitive inhibition, non-competitive inhibition and uncompetitive inhibition. This classification is important for you. Even enzyme inhibition is also essay question for you. First you have to copy this illustration. Okay, that is enzyme inhibition is of two types: reversible, irreversible. Reversible means here this type of inhibition is reversed. Okay, irreversible means it is irreversible, not reversed. Again, under the reversible you will have three types of inhibitions are there: competitive inhibition, non-competitive inhibition and uncompetitive inhibition. Then here irreversible inhibition, under irreversible only one type is there, that is suicide. Okay, suicide inhibition. Then coming to the first type, we are talking about the reversible inhibition. Reversible inhibition means here the inhibitor binds non-covalently with the enzyme. Okay, that means here the inhibitor is there that binds to the enzyme loosely. So enzyme inhibition can be reversed if the inhibitor is removed. So as it is binding loose to the enzyme, if we remove the inhibitor from the enzyme, this type of inhibition is reversed. And irreversible means here the inhibitor binds tightly, covalently to the active site and binds non-covalently but very tightly. Okay, that's what I think you are following. Reversible and irreversible. Reversible inhibition means here the inhibitor binds to the enzyme. Reversible means here the inhibitor binds to the enzyme inhibitor binds to the enzyme non-covalently. In the irreversible inhibition here, the inhibitor binds to the substrate, the inhibitor binds to the enzyme non-covalently, that is covalently tight. Sometimes it is not, it may be bind to the loose but very tightly. That means, what did you understand? In the irreversible inhibition, we can't remove the inhibitor from the enzyme. But in the reversible, the inhibitor can be removed from the enzyme. So here enzyme inhibition cannot be reversed if the inhibitor is removed. Then reversible inhibition is of three types. Okay, reversible inhibition means what here? Inhibitor binds to the enzyme non-covalently. If you remove the inhibitor, this type of inhibition is reversed. That is of three types: competitive, non-competitive and uncompetitive. There are three types of reversible inhibitions: competitive, non-competitive and uncompetitive. Then coming to the competitive inhibition, it's a competitive inhibitor, because of the close structural resemblance to the substrate, it binds to the active site and prevents the substrate binding to the enzyme. What's the reaction I asked you to remember? Enzyme plus substrate gives rise to enzyme substrate, that will further gives rise to enzyme plus product, right? So what is the reaction? Enzyme plus substrate to gives rise to enzyme substrate, which will further gives rise to enzyme plus product. That is the reaction. Here in the competitive inhibition, the inhibitor is there, the chemical substance. The structure of the inhibitor is almost similar with that of substrate. Okay, it is almost analog. The structure is almost similar with the substrate. Then what will happen in the presence of the inhibitor? Actually, on the enzyme, on the active site of the enzyme, who has to go and bind? Substrate. Normal reaction. But here, because of the similar structure, because of the duplicate, the structure of the inhibitor is almost same, the inhibitor go and bind to the active site of the enzyme and prevent the binding of the substrate. To form, in the presence of this inhibitor, there is enzyme inhibitor complex is formed. What is the normal one? Normal one is enzyme plus substrate to form enzyme substrate complex. But here what happened? Because of the structure, here the inhibitor has got structural resemblance with the substrate. Because of that, inhibitor go and binds to the active site of the enzyme and prevents the binding of the substrate. Okay, and prevents the binding of the substrate. So rather than forming enzyme substrate complex here, enzyme inhibitor complex is formed and there is no more production of the products. Okay, why it is called competitive inhibition means here, because of the structural similarity, there is a competition occur between the substrate and the inhibitor for the binding of the active site. Okay, because of the structural similarity, there is a competition occur between the substrate and inhibitor for the binding of the active site. That's what this is called, competitive inhibition. Okay, now here, now look at this picture. This is the substrate, this is the enzyme, and already I mentioned, no, always enzyme has a site for the binding of the substrate. So usually in the normal reaction, what will happen? This substrate will go and bind to the enzyme to form enzyme substrate complex right here, but that is a normal reaction. Okay, so first one, what happened? Green one is the substrate that binds to the enzyme to form enzyme substrate complex, okay, to form products. But here what happened? The inhibitor, in the competitive inhibition, substrate is there. Okay, inhibitor has the structural similarity with the substrate. Okay, this is the shape of the substrate, this is the shape of the inhibitor. Normally, if inhibitor is not there, what will happen? Nicely the substrate will go and bind to the enzyme to form enzyme substrate complex that will give rise to the products. But here what happened? There is another one is there, that is inhibitor is there. The shape of the inhibitor is almost similar like that of substrate. So here this shape is same and this also can fit into here. So there is a competition occur between the substrate and inhibitor for the binding of this enzyme. So what will happen here? Inhibitor, because of the structural similarity with the substrate, it go and bind to the enzyme and prevent the binding of the substrate. So here what is going to form? Enzyme inhibitor is going to form and there is no formation of the products. Okay, so that is the competitive inhibition. So competitive inhibition, the degree of the competitive inhibition depends upon the number of the rate of the competitive inhibition. It depends upon the number of the inhibitors present. If more substrates are there than the inhibitor, the competitive inhibition is normal, the rate of the reaction is normal. But if the okay, like this, okay, but there is, okay, but if the substrate concentration is less, inhibitor concentration is more, what will happen? The active site of the enzyme has been occupied by this enzymes, okay, by this substrates, inhibitors, okay, like that. And here, now that is the competitive inhibition. I hope you understood the competitive inhibition. Competitive inhibition means here, first enzyme has a site for the binding of the substrate to form enzyme substrate complex that gives rise to enzyme plus products. But in the competitive inhibition, there is an inhibitor which has the structure of the inhibitor is similar with that of substrate. Because of the structural similarity, instead of the substrate, the inhibitor will go and bind to the active site and forms enzyme inhibitor complex. There is no more formation of the products. But the rate of the competitive inhibition, it depends upon the availability of the substrates and the availability of the inhibitors. If more substrates are there, normal reaction will take place. If more inhibitors are there than the substrate, obviously inhibitors will go and bind to this one and then by inhibition, as there is C. Okay, as there is see here, this is the one, and the this is the competitive inhibition. Okay, this is the enzyme and this is the shape of the substrate. Actually this is supposed to go and sit here, but this is the shape of the inhibitor. If more inhibitors are there than the substrate, so rather than substrate, inhibitors will go and bind to here to form enzyme inhibitor complex. There is no formation of the products. Okay, actually if more substrates are there than the inhibitor, normal reaction will occur. Enzyme plus substrate to form enzyme substrate complex that further gives rise to enzyme plus products. Suppose if more inhibitors are there than the substrate, then obviously inhibitor will go and bind to the enzyme to form enzyme inhibitor complex and thereby prevents the formation of the products. Okay, that's it about the competitive inhibition. and here there is competitive, most of the drugs used for various treatments or their mechanism of action is based on the competitive inhibition. Example is dihydrofolate reductase. This is an enzyme. For this the substrate is dihydrofolic acid. Normally for this enzyme the substrate is dihydrofolic acid, but the competitive inhibitors for these two is aminopterin and amitriptyline. Aminopterin and amitriptyline, please note down, these two are the anti-cancer drugs. So these two drugs, what will happen, they will go and bind to the active site of this enzyme and thereby decreasing the folic acid production. As the folic acid is not there, thereby cell growth, cell multiplication is arrested. Thereby these two drugs are widely used in the treatment of the cancer. Please note down. And the next one is there is a succinylcholine. Succinylcholine, it has a structural similarity with the substrate acetylcholine. So in the presence of more succinylcholine, it binds to the active site of the acetylcholine esterase and prevents the binding of acetylcholine. So that's what it is used in the neurotransmitter. And next one, methanol. It's a competitive inhibitor. It has a structural similarity with the ethanol. So that's what there is the competition occur between these two for the binding of the active site of this alcohol dehydrogenase. So like that, most of the drugs in their mechanism of action, competitive inhibitors or competitive inhibition is the example. Then coming to the second one is non-competitive inhibition. So non-competitive inhibition means here inhibitor has no structural resemblance with the substrate. What about the competitive? In the competitive, this inhibitor structure is similar with that of substrate, but in the non-competitive, inhibitor is there but it has no structural resemblance with the substrate. Then inhibitor, so then where the inhibitor binds means it binds to the enzyme at the site other than the substrate binding site. So here enzyme is there. There is an enzyme, there is a specific site is there for the binding of the substrate. So as usual here enzyme will go and bind to the substrate, but here the inhibitor binds to the enzyme other than the active site. So inhibitor here inhibitor does not interfere with the enzyme substrate binding, but catalysis is prevented. Why? Because here as usual substrate will go and bind to the enzyme. But what happens? Here inhibitor binds to the other site of the enzyme, thereby forms enzyme substrate inhibitor complex. Usually what supposed to form to bring the catalysis? Enzyme substrate complex has to form. But here what happens? Substrate will go and bind to the enzyme. Along with that inhibitor also binds to the some other site of the enzyme to form enzyme substrate inhibitor complex, and thereby prevents the catalysis. Right here, look at this picture. This is an enzyme. This is the substrate. Normally what will happen? This will go and bind to here to form enzyme substrate complex. And that enzyme substrate complex supposed to give the enzyme plus products. But now what happened here? There is the inhibitor. So this inhibitor will bind to this is the active site for the binding. And this inhibitor binds to the other site other than the active site. There is another site on the enzyme. So inhibitor binds to the other site other than the active site. Here, so one site there is enzyme will be added, another site inhibitor will be added to form enzyme substrate inhibitor complex that prevents the catalysis. Right here in the first case competitive inhibition, there is a competition occur between the substrate and inhibitor for the binding of active site. But in the non-competitive inhibition, there is no competition between the substrate and the inhibitor, because substrate has its site for the binding, inhibitor also has its site for the binding. So ultimately, instead of enzyme substrate, here enzyme substrate inhibitor complex is formed that prevents the inhibition. So here look at this. This is an enzyme. This is substrate. So normally substrate will go and bind to the substrate binding site. And here normal inhibitor will have bind to the inhibitor, and then to form enzyme substrate inhibitor complex, there is no formation of the products. Now here, so here enzyme has two sites, one side for the binding of substrate, another side for the binding of the inhibitor. So instead of enzyme substrate complex, to form enzyme inhibitor complex, there is no formation of the products. That is non-competitive inhibition. Then coming to the uncompetitive inhibition. Uncompetitive inhibition means here inhibitor has no structural resemblance with the substrate, and inhibitor binds to the enzyme substrate complex thereby. So here enzyme inhibitor won't bind to the enzyme. As usual, substrate will go and bind to the enzyme to form enzyme substrate complex. Here inhibitor go and bind to the enzyme substrate complex to form enzyme substrate inhibitor complex. So increasing in the substrate concentration, that results in more enzyme substrate complexes that will inhibits the binding and therefore increases the inhibition. Okay, look at this slide here. This is the uncompetitive inhibition. Okay, right? Here inhibitor, it cannot bind to the enzyme, right? So substrate as usual, it can bind to the enzyme to form enzyme substrate complex. To the complex, then inhibitor will bind to form enzyme substrate inhibitor complex. Okay, so this one is called uncompetitive inhibition. Okay, and here because in the presence of the inhibitor, enzyme has no role. Okay, so like that, reversible means here the inhibitor binds loosely, non-covalently to the enzyme. That type of inhibition is called reversible inhibition. Reversible inhibition, you have three types are there: competitive, non-competitive, uncompetitive. Competitive means here the inhibitor is the substrate which will reduce the enzyme activity. That process is known as enzyme inhibition. But in the type of reversible inhibition, the inhibitors are bind loosely to the enzymes. So in the competitive inhibition, what happened? Inhibitor has structural resemblance with the substrate. So there is a competition occur between the substrate and inhibitor for the binding of the active site. Okay, and in the non-competitive inhibition, what will happen? The substrate binding to the active site, but inhibitor has another site on the enzyme to form enzyme inhibitor complex. Thereby there is no competition between the substrate and inhibitor here, but the catalysis is prevented. Okay, but in the uncompetitive inhibition, what happened? The inhibitor has no structural resemblance. It binds to the enzyme substrate complex and thereby to form enzyme substrate inhibitor complex. Okay, and thereby prevents the catalysis. This one is important essay question for you. Okay, and then coming to the next one is irreversible inhibition. Here the inhibitor binds tightly to the enzyme. This can be explained by suicide inhibition. Suicide inhibition means here the substrate analogs are transformed into inhibitors by the enzyme. The substrate where it is supposed to bind to the enzyme, that itself modified into the inhibitor. Okay, the substrate itself is modified to inhibitor. No other inhibitor is required here. The substrate whatever is supposed to join to the inhibitor enzyme, that only modified into the inhibitor. So the products of the reaction is highly, so sometimes the products which are formed here are highly reactive and inhibits the enzyme action. So its own killing. So that's what here it is called suicide inhibition. And some examples of irreversible inhibition with the medical relevance: aspirin is the drug. Okay, its target enzyme is cyclooxygenase. Mainly it is, what is aspirin? Anti-inflammatory and antiplatelet activity. Its mechanism of action is irreversible. And you can take the penicillin, it is an antibiotic. It is also irreversible and it can target enzyme is transpeptidase. And there is allopurinol, it is a drug widely used in the treatment of the gout. Okay, and it is also irreversible suicide inhibitor. It inhibits enzyme xanthine oxidase. Okay, like that, that's it about the enzyme inhibitions. Okay, so enzyme inhibitions means concentrate on two types. Okay, enzyme inhibition means this is the process which which decreases the interfere the enzyme activity, and the inhibition is brought about in the presence of inhibitor. Inhibitor is a substance. So there are two types of inhibitions: reversible, irreversible. Reversible can be explained by competitive, non-competitive, uncompetitive, and the irreversible can be explained by suicidal inhibition. Okay, that's it about this one. So the major topics we have finished here in the enzyme so far is enzyme classification, factors affecting enzyme action, and the next one is rate of enzyme inhibition. Okay, and one small topic is left over, that is enzyme isoenzymes. Let me finish off that one. With that, this chapter is going to finish. Okay, so with this, enzyme is finished, and give me 1 minute time, I'll change the slides for isoenzymes.